Good morning, good afternoon, and good evening to everyone. We're just going to give it one more minute to allow for a few more to trickle in. We've got around just over 100 people who have committed to attending live, and of course, we'll have it available afterwards as well. So just bear with us for one more minute. Thank you. All right, given the jam-packed agenda, we're going to get going. And for those of you who are joining later, again, we'll have this available on YouTube after the webinar is complete. So thank you all for joining, and what will be an undoubtedly really exciting presentation. I'm really looking forward to it. First, we must acknowledge that the land we are meeting on is the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat people, and is now home to many diverse First Nations, Inuit, and Métis. We also acknowledge that Toronto is covered by Treaty 13 with the Mississaugas of the Credit. In terms of housekeeping rules for today, there is a live transcript available, so please turn on the feature if you wish to access it. It's available on the bottom menu. Please also ask questions in the chat box, and we will make sure that they are answered appropriately throughout the presentation. This is also being live streamed and therefore recorded, as you may have noticed, and it will be available on YouTube afterwards. So the link to the channel is in the chat box as well. So without further ado, I would love to introduce our speakers for today, Dr. Steve Gardner and Naheed Kurji. And I will pass it over to Steve, who will start sharing, and they'll do some introductions. So thanks very much, and let's get on with the show, shall we?
Excellent. Well, thank you, Jen, for the quick introduction, as well as the acknowledgement of the land that we in Toronto and Canada are on, though admittedly with Steve in the UK, I saw him smiling. This is a virtual session, and I'm sure a lot of people are there globally. So I appreciate all of your time as we respect the land that we in Canada are on. Very excited about this opportunity. Steve, if you can go back just a couple slides, please, to introduce you to the second session in our Molecule to Medicine series. This is a series that Cyclica has initiated, but that we are bringing together with a number of our partners and thought leaders in the space. And today, we have an exciting conversation on disease mechanism and target discovery. If you can go to slide 3. As part of this discussion today, Dr. Steve Gardner, co-founder, CEO of PrecisionLife and one of Cyclica's business partners, will be walking you through the topic that I just mentioned. And given his background and experience in the space, it is set up to be an informative, invigorating conversation in a very exciting space, which is target identification for the discovery of medicines. This is a really hot space, and you'll hear today about the economic hurdles of misprioritizing targets in drug discovery. Next slide, please. And our background at Cyclica is we are an AI and drug discovery company by what has been framed in the marketplace. But I'd like to say that we are much more than just an AI and drug discovery company. We call ourselves a neo-biotech, the new form of a biotechnology company that is computationally focused, but with a deep empathy on the complexities of chemistry, biology, and the requirements of advancing molecules to medicine. And as part of our vision as a company to advance the biotech pipeline of the future, the largest, most therapeutically diverse pipeline of assets in the pharmaceutical industry, we recognize that we can't do this alone. collaboration with companies that have unique and state-of-the-art technologies that are adjacent to what we're doing, like with what PrecisionLife is going to explain to you in upfront target identification, biomarker development, and patient stratification strategies, is core to Cyclica's ethos. Let's all come together so that we can advance molecules to medicines for patients who are desperately awaiting. And so with that, I will pass it over to Steve to take it from there, and I'll jump in for any questions or comments as they come through from the audience. Thank you, Steve, take it away.
Thanks so much, Naheed. That's a great introduction, and it's a great place to start in terms of why we need a new biotech ethos and why we need a new biotech pipeline for the future. And I think a lot of it comes because the traditional blockbuster model of medicine has really been less cost effective, more inefficient over the last decade than we really need. And we're finding that one size fits all medicine, and particularly a poor understanding of which disease biology is relevant to which patients, is bad for everybody in the ecosystem. It's bad for patients, obviously, because many times they're getting drugs that don't actually work for them. And many of the most prescribed medicines are actually effective only for a minority of patients. It's bad for healthcare systems because that creates wastage. You know, they're prescribing expensive but ineffective medicines and having to deal both with patients whose needs are not being served and also over medication of those patients. And I'd argue it's also bad for pharma. And we've seen productivity declines significantly. And I'm going to drill into that a little bit more. So there's really two things going on with pharma productivity and the return on investment of pharma R&D, which has reached record lows. And partly this is because we're seeing a lot of late stage failure. You know, the cost of drugs, developing a drug is going up, as we all know. We've all seen those headlines. But a lot of that is because of late stage failures, particularly in phase 3 for efficacy. And, you know, efficacy is about not knowing which patients to put in your trial population. And I'll pick on something like Alzheimer's. There have been 130 straight failed clinical trials in Alzheimer's. And it's not because the pharma companies didn't know what they were doing. It's not because the molecules are not tailored to a particular target. But it's because it's really hard to put the right patients into a clinical trial who are going to benefit from that particular potential medicine. And that has consequences. It has consequences for late stage failure, as I said, but also because when you come to prescribe those medicines, you've got to be confident as a healthcare system that you can justify the cost of them. And when we look at Alzheimer's in more detail, and I'll use this as a case study later, the highlight that you can see is that we have actually a really strong case for stratifying that patient population at a much more detailed level, using all of the information that's available about those patient populations and getting under the skin of that disease to dissect it into specific subgroups that are going to benefit from new types of medicines. And that creates opportunities for novel target discovery around particular disease mechanisms. So you might say, well, you know, I thought this was going to be a solved problem. 20 years ago, we had the Human Genome Project, and you know, there's a little highline here that says we've got a new medical era coming. And of course, you've got to remember that when we instigated that program, our view of disease was very different. It was driven by things like monogenic diseases like sickle cell, where we can see, you know, single base mutations. This AT going to a TA leads to a glutamic acid changing to a valine, which creates a lovely little sticky patch on the outside of a hemoglobin molecule, which causes, when these are deoxygenated, them to form little fibrils. And those fibrils cause the red cells to sickle, and they reduce the transport of oxygen. It's a really direct mechanism. Change in the DNA leads to a change in the protein, which leads to a structural change and therefore a loss of function. And that's how we used to view diseases. And we thought that when we'd done the Human Genome Project, we'd be able to collect all of those. And that's really where the analytics came from. So genome-wide association studies, which are used to analyze large-scale patient data, are really looking for those features. They're looking for genetic signatures that occur in a load of cases and very few controls, and which stand out with what we call a large effect size. In other words, they're very, very significant when you evaluate the mutation and its occurrence in patients versus controls. Unfortunately, that means that things which are less frequent don't show up as much. So if you have something that's relevant just to a subgroup in your patient population, you're not actually going to find it using standard genome-wide association studies. And this limits what we've been able to do with the patient data that we've been collecting over the last 20 years or so. There are some other challenges involved as well. You have to be very sure that the patients you're collecting have the same diagnosis. Really, the causes of their disease are actually genuinely the same. And also that the disease is not caused by different factors in different subsets of patients. The bottom line is that this really works well in somatic cancers and also in rare diseases. And those are the areas in which we've seen the biggest progress to date using genomic analysis. Where they tend not to work so well is in chronic diseases. And chronic diseases are a huge burden, socioeconomic burden, a disease burden for patients. They cost about 80% of the $10 trillion or so that is spent on healthcare to date. And there are some huge areas. Diabetes, it's 15% of the US health spend, which is 18% of GDP. Put that into context, nearly 3% of US GDP is spent treating diabetes. I mean, that's a crazy number. That is a huge number. And it's growing faster than the GDP is. Globally, only about 20% of diabetes sufferers are over 65. Naheed, you had a question?
Keep going. I'll jump in with a question momentarily.
So there is a demographic time bomb coming, and it's not just diabetes, it's dementia, it's cardiovascular disease, respiratory disease, autoimmune. They're all growing massively. And here's the fundamental problem. They don't work the same way as those monogenic diseases. If you look at something, for example, take bipolar, you get a diagnosis of bipolar, you're allocated to one or other ICD-10 code, probably because the clinician has observed some symptoms and felt that fits you into a particular category. Or perhaps they know what is reimbursable and therefore they've sort of shoehorned you into that kind of category. But you're going to be on a clinical pathway based on your ICD-10 code. So you're pretty much going to get given one or other of the classically diagnosed prescribed medicines. The problem is that bipolar is a spectrum disorder. Like all chronic diseases, it's not caused by a single genetic change. It's caused by multiple interactions. And so what we actually have are an overlapping set of patient populations, subgroups, where the causes of those diseases, the severities, the progression rates, the symptoms, and the treatment responses are going to be different for each of these patient subgroups. And so that creates a real challenge for the kind of analytics that we use and a real opportunity for novel target discovery in this space.
So Steve, I have one question and one comment for you. And I think the past few slides, past 11 slides, have set the stage up for the problem and the opportunity. And if we come back to this example in bipolar, it reminds me of something that may be more salient to the audience and to people generally in the public, which is in depression, where depression is not really well understood. Different people experience depression very differently, but generally when they go to the physician or to their family doctor and they're diagnosed with a form of depression, they're generally treated with the standard of care that may not be relevant for that individual. And I've read many cases and gone through experiences where to get to the ideal medication for that N of 1 could take anywhere from 10 to 15 different attempts that each take 4 to 5 months to figure out that the first 9 were not the right one. And so I have, on the back of that observation, I have 2 questions. Number 1, is this generally an issue in CNS related diseases? We're talking about bipolar, I've just talked about depression. Is the patient stratification strategy more difficult when it comes to those more complex diseases, polygenic diseases, many of which are CNS? Question 1. Question 2, if you look at the past 8 to 9 slides, you laid out the economic challenges that have existed, pervaded the industry for a long time, and productivity is decreasing, costs are increasing, failure rates are increasing. And then there is the advent of, or the kind of the work that emanated from the Human Genome Project, but that's been 20 years. So why has it taken us 20 years to start to see a clip up in productivity? What are the big lessons that we've learned, not just philosophically on the approach and the reductionist approach to drug discovery, but what have we learned about data and new methodologies and combinatorial technologies to advance better targets?
Yeah, great question. So on the second point, you're absolutely right. You know, I think it's taken us a long time to understand just how messy biology is and just how disparate some of these really complex chronic diseases are. You know, because we had a tool like genome-wide association studies, and that was the available compute could actually run that tool. That was the one that we chased those diseases with, and it took us a long time to figure out that we weren't going to find all of the signal that was relevant in those diseases, and that has, you know, that has a lot of profound consequences in terms of where we are now with new developments in AI technologies, much better computational platforms to actually analyze these data, but also the cost of collecting those data and the quality with which you can collect has improved massively over the last decade or so. On the second point, yeah, I mean, there's a whole class of diseases. So certainly CNS is an absolute poster child for complexity, as is, you know, not just psychiatric disease, but also neurodegeneration. But even now, you know, things like cardiovascular disease, bits of it are genetic and bits of it are monogenic, sorry, and other bits of it are not. Other bits of it rely on this combination of features. Things like endometriosis, you know, autoimmune disease, the respiratory disease, asthma, you know, there are huge factors at work and unmet pools of patient need that are just not found using the existing genomic technologies. So I think that's why we really need, as you'll see in a second, why we really need a whole new way of looking at disease and analyzing these large-scale patient populations.
And before you jump into the next slides, there's been a lot of work by many companies, both large and small and academic institutions, on understanding disease mechanism and identifying targets that are related to that disease. And there's been work on open sourcing a wealth of data, and one of the, I guess, open source databases is on open targets. Can you talk about why that open source capability, while it's been helpful for open science, why it hasn't yielded the results and productivity still declined, and what are the biggest opportunities that you and Precision Life have contemplated, and how that your strategy is a bit different than just identifying targets for disease? And perhaps the slides will go into that. I don't want to jump ahead, but I think that would be an important point.
Yeah, I mean, Open Targets is a fantastic resource, and we absolutely do use it. It's a great collation of what is known around potential targets. It's a great summation of the existing research that's out there. But what it doesn't do is take a patient population, such as the one I have behind me, which is schizophrenia, and analyze hypothesis-free and in an unbiased way, what are all of the combinations of features that are associated with disease within the patient subgroups here. So it doesn't tell you anything about what's going to work for one subgroup versus another subgroup. It doesn't, it reports what's in the literature, but it doesn't find new associations because you've got a large-scale patient data set that you can be working from. So it's very useful, as are many other resources, and I'll reference a lot of them along the way here. But it is only one way of thinking about it. It isn't really a novel analytics tool for getting into patient data and using it for that primary target discovery piece. Let me show you what we do, which will go some way to answering those. So a standard genome-wide association, as I said, finds one thing at a time. And the major problem with that is because diseases are multifactorial, the biology that drives them contains feedback loops. And those feedback loops, some of them inhibit each other, some of them amplify each other's effects. And these are complex interactions of genetic and metabolic networks. And you can't capture that signal using a standard GWAS. You need to be able to look at combinations of features and how they associate with particular disease. And that's what we do. So this combinatorial approach allows you to look, this is a real example. This is a 6 SNP signature that occurs in 150 women who have pathogenic BRCA2 variants, but who don't get breast cancer. So they would have been told that they've got a risk of 60 to 80% of developing breast and ovarian cancer in their lifetime. And yet these patients don't. So what you've got there is, it's a component of a much more accurate risk score. It's a patient stratification biomarker. And it also gives you an opportunity to create a genetic explanation, a testable mechanism of action hypothesis about which of these genes is actually driving that observation. And this is what we rely on. This is what we build out our tools from. And if you're a little bit, if you don't understand why combinations are really important, I have a simple analogy. I hope it's simple. And this is poker. GWAS is a little bit like doing a big analysis of loads and loads of hands of poker and coming up with the insight that hands that contain aces, kings, queens, and jacks more usually win than those that contain low value cards. And so you make a prediction that anything with an ace in it is going to be a really great hand. Of course, we know in poker, and the same is true in biology, that it is the combination of cards that will actually determine what is a winning hand. So here, a straight flush with relatively low value cards is the winning hand. What I'm getting to is that individual features with high odds ratios in a GWAS don't necessarily make the best targets. They're not necessarily even causally related to the mechanism of the disease. So we use this combinatorial approach. It looks for multiple features in combination. It allows us to find a lot more signal than you can find using GWAS. And it also allows you to segregate patient populations into subgroups. And that's precisely what you need to address chronic disease and the germline risks associated with specific cancers. How much more signal? Well, we did a meta-analysis of the factors driving severe COVID. And, obviously this has occupied a lot of attention over the last few months. Various groups have tried across millions of patients to assemble large-scale patient populations and then run genome-wide association studies. Couple highlighted here, up to 2 million patients, and they found 11 loci associated with one gene or 13 loci associated with about 6 or 9 genes. But these really don't explain the diverse symptomology that we see, the mechanisms at play inside COVID, and why it leads to all of these wildly different patient responses, or the range of symptoms that we see. Before that, using combinatorial analysis, we'd found 15 months ago, we published a paper on this. We'd found just from a few hundred patients, many more loci. We found 6 mechanisms that get not just into viral replication and binding and cytokine cascades, but also into some of the metabolic signatures associated with severe COVID. And that led us to druggable targets. It led us to validating this. We found known active chemistry against these targets, that one of the inhibitors of a target actually demonstrates some of the most potent anti-SARS-CoV-2 activity ever seen in a viral plaque assay. And we also validated it in clinical data with United Healthcare on their 300,000 patient portal. So we predicted signals in the genetics that would be seen in the longitudinal claims and lab data that in fact did turn out to predict who was most at risk of needing ventilation or having severe disease. So there really is a lot more signal in the genomics than we're able to find using the current analytics. And this can be applied across a very wide range of diseases. To your point earlier, what are the chronic diseases that this will benefit? Well, we've run 26 projects in the last 18 months or so, back to your need for a scalable pipeline, and across a very broad range of these disease areas. Now, I'm going to use Alzheimer's as my case study here because it's pretty topical, and I'm going to take you through what we actually do in this process. So we start off with multimodal patient data. That's just a fancy way of saying, we have some genomics, we have phenotypic data, we have clinical information, and probably some epidemiological and environmental data. We run the combinatorial analysis, we find these disease signatures, and they contain references to SNPs. Well, those SNPs can be mapped to genes, and then we can evaluate by annotating those with a lot of information from places like Open Targets and others. We can annotate all of that and then decide which of those are going to be the most useful to take into a target validation process. And in many cases, we'll have some known active chemistry, which can be a tool compound, a starting point for this. And we can get into in vitro or in vivo testing very quickly. So if I apply that to Alzheimer's, here's a study we ran on UK Biobank, about 900 cases, 1,800 controls. It's a genotype only study. If you run that through GWAS, you find the ApoE signature. That's fine. That's pretty much what you would expect. When we run that same study, we find 267 loci or SNPs, and we can cluster those on the basis of the patients in which they co-occur. And what this allows us to do is to start to see those communities, those subgroups of patients in which share a common cause of their disease, or at least a common association with their disease. And when we start to look at the targets that represents, you start to see not just APOC1 and ApoE, which are the biggest signal, but you also see a whole bunch of other mechanisms coming into play. So you're doing two things. Number one, you're opening up a whole bunch of new targets. And number two, you're finding the patients which those can be associated with. And by definition, patient stratification biomarkers. So what we did further on this is to look at these communities and see how similar they are in terms of the mechanisms, the genes, the pathways that are involved. And we got a really stunningly clear stratification of this. So we have a patient subgroup for whom lipoprotein metabolism and defects in it are related to their disease. But we have other groups where a prior viral infection and an aberrant innate immune response seems to be associated with the disease. Another group with dysfunctional autophagy, another group that can't maintain copper ion homeostasis, another one that goes through metabolic syndrome from obesity into diabetes and then into Alzheimer's. And then a final one, which is a recognized clinical endotype, where there's more manic presentation because of serotonin dysregulation. So here, I mentioned 130 straight clinical trial failures. If you're chasing lipoprotein and all you're doing is basically you're addressing about a third of the patient population, but you've included all of these others, however beautifully tailored your molecule is to that particular mechanism, it's going to be really hard to demonstrate that signal. Plus, you're missing a whole bunch of opportunities for novel targets that address other aspects of the disease or other causes of the disease for different subpopulations.
Steve, I'd like to jump in here with two questions, both of which are a little bit higher level, to take a step back. So you're walking us through how you can layer on a bunch of omics data to stratify patients and identify novel mechanisms of disease and potential targets. First question is, with that list of potentially novel mechanisms, novel targets for a specific patient population, how do scientists then prioritize their targets for which to then progress the development of drugs? You can't go after everything, especially early stage biotech companies or those with limited resources. So then how do you go about prioritization? That'll be question number one. And then can you talk about animal models? We're talking a lot about patients, but to then get to the clinic and get into patients, we have to, in the current way of doing stuff, is test in animals. And as there's a lack of animal models in Alzheimer's disease, how can target discovery and even in silico drug discovery help in finding drugs for AD?
No, it's the latter. The first part of the question I'll answer in the next couple of slides, so I'll defer that one if that's okay. But the second piece is a really important area, and it comes to a whole raft of new technologies that are available. You're absolutely right. Particularly in CNS, the animal models are just not great. Particularly in neurodegenerative disease, they're really lacking. Some of the ways that we go about doing that, we will use iPSCs, so patient-derived stem cells from patients with the particular condition, and ideally we would target those at specific subgroups of patients, so you can be very specific about the mechanism you're trying to target. Organoids and other other patient-derived tissue systems are a really useful tool that are starting to become much more prevalent inside this, even inside CNS. And you're starting to see 2D arrays on multi-electrodes. You're starting to see organoids, which are 3D architectures, even of brain tissue or mixes of motor neurons, astrocytes, and microglia, for example. So it is becoming possible to do those. You then need to, there is then a translation into the rodent models or other models, which is necessary. But I think what we will see is that moving to first in man and actually doing some studies earlier in man is going to be pretty pivotal in the development of a lot of new therapies in the CNS area.
Excellent. Thank you for that. And there's one question that came in from the audience. Your slides may address it. If not, here it is. How do you develop biomarkers for diseases and disorders that are difficult to define?
No, that's a great question. I mean, the short answer to that is that each of these patient subgroups is, if I just go back a touch, each of these patient subgroups is connected to one of these sets of SNPs, and those SNPs become that defining criteria. You can use them as inclusion and exclusion criteria during clinical trials design and development, or you could potentially use them for diagnostics in a healthcare setting. But these features are the things that we consider to be the most useful points of developing a new biomarker. So, I mean, just taking just taking your first question earlier. So this is, we basically run a cascade process. So you've just seen, I said we have 113 genes associated with Alzheimer's. That doesn't mean that they're all causal. It doesn't mean that they're all drug targets. It just means that they're associated. That's fine. So now we need to take those gene associations and turn them into a list of potential targets. And we do this by analyzing all of that annotation data, the open targets plus tissue expression plus a whole bunch of other things. And we run a whole series of heuristics that I'll describe in a second. We then get to a point where we have to know whether these are druggable, use it, particularly whether they have a therapeutic product profile that we're going to be interested in. In respiratory, the holy grail is, could I give a once a day oral therapy, for example, as opposed to inhaled compounds? Do we have the right dosing? Is the modality appropriate and the delivery mechanism appropriate? And that will allow us to then make really detailed decisions about which ones we want to take forward into testing and into novel chemistry design. But if I put a bit of detail on that, we're informed by a whole range of functional characteristics that are known about those targets. So this is exactly what's in open targets and beyond. All of these interactions, the known metabolism, interaction factors, what the potential consequences of interacting at a specific target are in terms of its control of other metabolic processes and pathways. So we collate all of that information. We do it not just at an individual target, but it's also important to realize that there are many ways of modulating a particular target's impact in the metabolism. You may go upstream, and that may lead you into a different modality. You may go slightly downstream because this is a big control hub and you have an opportunity to have a more specific effect at a different point in that pathway. Or you may start to affect things like transcription factors rather than looking at changing directly or compensating directly for a loss of function mutation in a particular gene. You might look at its expression and how active that particular pathway is inside a disease state and make changes to upregulate or downregulate that. So there's lots of different points at which you can interact. AstraZeneca, GSK, AbbVie have all published on frameworks that they use for thinking about what makes a good drug target. And, AstraZeneca have the 5 R's of drug discovery. And this basically says, if we pick the right target with a genetic association, with a plausible mechanism of action for affecting the disease phenotype, and we can find that target in the right patients because we've got biomarkers to select responders, and we know that that target is differentially expressed in tissues that are associated with the disease, and we know that the compounds that act at that target have good bioavailability and a decent safety profile, and of course that we've chosen our therapeutic product profile with a good, we've got freedom to operate, we've got unmet medical need, we've got clinical relevance in terms of frequency in the population. That can increase, that has been shown by AstraZeneca to have increased their likelihood of going from a project initiation through to a successful phase 3 by almost 5 times. And this is one of the great things that you can do, but it relies on you having all of this great information. Now, we use all of this, and we also use a 6th R, which is the potential to be able to demonstrate efficacy. So back to that original phase 3 failure issue that we had. This is all about mapping the influences on your chosen target's biology to patient subgroups and being able to identify what's actually going to work and which targets are you going to be able to build a clinical trial population that allows you to demonstrate efficacy. And so, when we look at these communities of patients which are connected to subgroups of, so these communities of SNPs which are connected to subgroups of patients, we're really doing a scoring system to help prioritize that feature. Because genes they might affect 5 of these different patient populations, or they may just affect 1. That's going to have a pretty decent impact on your chances of developing downstream a medicine. So we bring that into the mix, but it's dependent on having this this view of these level of insights into what's driving disease biology and how it maps to your patient populations. Of course, there's another piece to this, which is the druggability, and this is actually a set of results from one of our collaborations around using ligand design, I think in this case, to evaluate the polypharmacology of a particular target and to evaluate the likelihood of getting chemistry in this case that has the right ADMET profile. It has the right selectivity and affinity that we're interested in. It misses hERG, but in this case, it hits the blood-brain barrier penetrance markers that we're interested in. And you can add heuristics like this into any of those processes that we use for prioritizing the targets. Where that's led us to is, getting down to that short list of targets that we then want to put into testing, and having a matrix, a red, green, amber, red, amber, green, even set of criteria across a whole bunch of things that we really care about. And if we have too many red flags in here, then we just don't choose that. We don't prioritize that. It's interesting, in our interactions with customers, this level of novelty and how much supporting evidence is a real mixed bag. Some people want really, really innovative stuff that is first in class and never been seen before, but plausibly has a mechanism of action connection. Other people want to see more supporting evidence, and that goes back to, does it appear in open targets? Has it been tested in another inflammatory disease? And we're going to use it in this kind of indication. But those are really the thought processes that we use and the levels of evidence that we bring into the mix.
I think, Steve, what's interesting there, if we just take a step back and look at essentially the proteome, 20,000 known proteins, you look at those drugged by the pharma industry, can be anywhere between 8 and 1,500 that the entire industry has been focused on for a very long time. And there's the balance, if you deduct kind of the intrinsically disordered proteins and the ones that are more difficult, there's huge opportunity outside of those 800 to 1,500 that are generally novel, or they're low data but high opportunity. And you're seeing, at least I'd love to get your perspective, what I'm seeing, what we're seeing in Cyclica, is that increasingly more pharma companies, more emerging biotech companies, and even more academic and early stage biotech companies are going after novelty versus a follow-on strategy for target selection. Are you seeing that as well?
Yeah, it's a little bit of a mixed bag, as I say. I think that is generally true. I think there's a real understanding that certain target classes have either been mined out or simply are not working as well as we would hope that they do. And so there is certainly a very distinct appetite for a lot of novelty. But then, there are still some slightly more conservative voices around as well who appreciate having a lot of supporting evidence, particularly since these target discovery methods are new, and we haven't completely solved disease biology at this stage. It's not there's no level of certainty here. You're still, if you're going to commit resources to doing that validation, you still want some level of supporting evidence. So it is a mixed bag.
So given your background, and for those that don't know, Steve, as now CEO of PrecisionLife, has a rich background in the pharma industry. And so you've been on the other end. you've listened to companies like PrecisionLife saying, I have these technologies. So let's put your pharma hat on for a second. If today in 2021, a company were to come to you with level of novelty, very highly novel versus more common, but here's another way of thinking about it, what would you gravitate towards?
Oh, the impossible question. Yeah, it is a slightly impossible question. Frankly, I think if you've got a patient stratification biomarker and you can find a really interesting unmet medical need, I would be, personally, hey, I'm a startup CEO. I'm a risk taker. I would go for innovation all day long because I think
And
there's
I think
more
it also
opportunity.
depends on it depends on the disease, depends on the opportunity, the market opportunity. So I'll get you off the hot seat with an impossible question, put something right down the middle for you. A question from the audience. Really great presentation so far. So thank you on behalf of Steve and myself for that comment. Can you comment on Biogen's aducanumab as it relates to numerous failed on Alzheimer's clinical trials? What's happening with that now in the market? And then there's one more question that has come from the audience that I will ask after you answer that, Steve.
Okay. Well, I don't know the reality of this. I'm not on the inside of it. I read what everybody else reads in the press. So I go by that, and I know what we've found about Alzheimer's, and I draw inferences not specifically about Biogen, but about the wider industry chasing the amyloid and tau hypothesis, and where that is going to take us. I don't think it tells the whole story, as I've suggested. I think the bigger challenge as we reflect on that is I did see a report I think it was in Stat News, that the level of prescription around that compound is actually pretty low right now. And I think that goes to my point earlier about the forecast peak sales being hard to achieve if you're not able to put a tool into the clinician's hands that says these patients will benefit from this drug, which I want to charge $56,000 a year for. Unless you give them tools that can inspire that confidence, it is going to be challenging. And particularly as we move to a more outcomes-based remuneration model inside the industry, I think it's going to be advantageous for pharma companies not just to use these patient stratification biomarkers through the development process, but actually at product launch, to use them as tools to engage the marketplace and encourage prescription, because you're increasing the responder rate. And I think from a pharmacoeconomic perspective, that's actually a really advantageous thing for a pharma company to do.
Thanks for that. We have 11 more minutes. There's one more question, and Steve, I'll let you finish off with your presentation. The question comes from Amrit Singh. So thank you, Amrit, for engaging. Given that many complex diseases also have comorbidities, example, heart failure, what are your thoughts on a polypharmacy approach to treat diseases? Example, test target combination 1 + 3 + 5 over a different combination. I'm going to jump in and take that quick answer, and then Steve, I'd love to hear your perspective. Given that at Cyclica, we've been talking about polypharmacology and the importance of harnessing polypharmacology into drug design for a long time. We know that molecules interact with anywhere from a dozen to up to 300 unanticipated off-target interactions when placed into a complex biological system. Professor Maria Bolognesi from the University of Bologna published a nice paper back in, 2014-15 timeframe, where she said complex diseases like CNS diseases are often impacted best when you take a multi-targeted approach. And then there's some work by Morphy and Rankovich, and then a group at Novartis and GSK with academic works, and it's a paper called Recognizing the Potential of In Silico Approaches to Support Multi-Targeted Drug Design. And so what's become much more fashionable, I'd say academically, theoretically, for the past 10 to 12 years, is that looking at combinatorics, looking at a pathway, multiple targets for a given disease is the best way of going about it, especially for diseases where multiple targets are understood to have the phenotypic or biological event. Going after that single target approach and not considering unanticipated off targets or the multiple targets on a pathway that one should perturb is a suboptimal strategy, at least in Cyclica's mind. And so bringing combinatorics into play, thinking about a pathway approach and therefore a panel approach to drug design is key. But I'll also say, and then I'll pass it over to Steve, is designing molecules for one target of interest while avoiding others for selectivity reasons, or multiple targets and avoiding undesirable off targets that are linked to toxicity is only answering the one question. The next question that one needs to ask is, are they synthetically feasible? Are these drug-like in nature? Do they have the right ADME properties? And especially if we're going after complex diseases in the brain, does it pass the blood-brain barrier? Does it avoid hERG liability? And so taking a much more holistic approach with polypharmacology as the center, in our view, is critically important. This is not a Cyclica presentation, but the conversation was on polypharmacy, so I wanted to take that stab. Steve, what do you have to say?
Yeah, and I mean, to be honest, I agree with everything you just said, Naheed. I think it is incredibly important. I don't think, as we've just shown with Alzheimer's, the interesting thing about that Alzheimer's graph that I showed you when we got to the number of patient populations is that 70% of patients actually have more than one of these signatures. So this talks to the spectrum nature of disease. It talks to why, they've got a little bit of lipoprotein going wrong, but they may also have some autophagy defects. And you are going to need more than one way of addressing that. And, whether that's, there's a variety of different ways of thinking about polypharmacology in that perspective. I totally agree that you've got to get, you've got to be able to recognize what's driving disease within a large set of patients, and then you can start to design things that have more combination effects. What I will say is it's an awful lot harder to test combinations than it is to test individual compounds, and so the polypharmacology does bring other issues into the mix that make the job of designing molecules much harder, as well as figuring out what the biology is that's going on.
And I think even going back to one of your examples where you have mutant targets versus wild type, and you want to design for the mutant target while sparing the wild type, like that's where a polypharmacy approach can play an impact. And Emery, if you're interested, there's some stuff on our website that we talk about and some EGFR mutant work that we've done. So Steve, we have 5 minutes left. And there is one more question that I'd like to get to round it out, but back to you.
Okay, well, I'll finish off really quick. I was just going to say that our output here is really the input into the next phase of this series of lectures or webinars. This is where it enters the testing phase, where we get into patient-derived cellular assays, where we start to get into non-pharmacological ways of phenotypic screening to evaluate whether the target actually does have disease modification potential. And then you're into probably more biochemical assays, as well as these type of assays that support a design make test cycle, into in vivo testing. But I'm not going to talk about any of that because A, I don't have time, and B, they're the subject of other speakers. So what I did want to touch on was just some of the other opportunities here. So this is a map of ALS. And right at the heart, you can see those patient subgroups with the SNPs that are driving disease. And we can map those to genes, we can map them to pathways, and we can map them to all of the known active chemistry. So again, this is the open targets kind of, or drug bank, Chembl type approach. But critically, what we've got here that those resources don't have are patients. So where the subgroups are, what's relevant to that set of patients, and that allows you to do a couple of different things. It's a treasure map for where do I start with picking the targets to go after. But it also, when we've done 26 disease projects in the last 18 months, that then becomes a resource to say, when I've got a molecule that works at this target, what else is it going to be good for? Can I see that target is relevant in a clinically significant patient population? And can I then predict that I actually have a label extension or a repositioning opportunity with that particular target, and really track the biology back across multiple diseases? And I think that's going to be incredibly powerful. And I'm just going to finish up with this final insight, with this final thought, which is that the insights that we generate have a lot of different applications. As I've said, I've talked about novel targets, I've talked about clinical trials design and diagnostics, I've mentioned some of the applications in healthcare. But this is a virtuous circle. As we start to have useful insights coming out of these disease studies, it really starts to increase the value of healthcare data collection. And that becomes a resource that we can use for future studies. And, I'm really excited about the opportunity to collect in routine care more richer patient data sets and use those for further studies.
Excellent. Steve, are you done?
I'm done. That's
wonderful. Me. Great presentation. Fantastic conversation. Appreciate you letting me interrupt with a few questions on my mind and a number that have come through on the Zoom chat. I will say there's 2 more questions that have come through. I will note for anybody who's listening, anybody that cares, my team is giving me a hard time on that polypharmacology question, thinking that I purposely asked that question to then answer that question. Absolutely not. Amrit asked that question. I have no idea who Amrit Singh is, but wonderful question. And Amrit is laughing out loud right now. So thank you for giving us a softball down the middle, but that was not placed. So Steve, 2 last questions. How has the human genome sequence accelerated the drug discovery process? What have been the big impacts? Question 1 and question 2, and then I'll close out. GWAS versus combinatorial analysis. Which one to consider in identifying targets, considering the pros and cons? Then the last one is a bigger question, but go away.
So I think that the Human Genome Project has had some very significant, has led to some very significant advances, primarily in idiopathic rare disease and in cancers. I think its impact across other disease areas has been much less marked and has been actually quite, if you asked us 20 years ago when we were kicking all of this off, where we would be now, I think we thought we would be further forward. So then, combinatorial analytics, I think, gives us another set of tools to find another level of signal that is more appropriate for some of these diseases. And it's an and, it's not an or, it's an and. To allow us to analyze, chronic diseases and find patient subgroups where we really can find novel therapies. And I think that is one of the huge opportunities that's out there now. And then did I miss a final bit of that question?
I think that's about right. And we're at the time.
Okay.
So Steve and the entire team at PrecisionLife, who've been working behind the scenes, Noah, Rowan, and the team, always a pleasure to work with you guys on this. Thank you so much for dedicating your time to this open initiative to inspire, to educate, to provide perspectives to a very interesting group of individuals who are clearly very interested in what the future looks like and how medicines are being brought to patients. So this was episode 2, disease mechanism and target discovery. As I mentioned at the early end of this presentation, where we showed the series roadmap, the next set will be on target identification and target deconvolution, an important topic coming out of phenotypic screening, where you have molecules and you have biology understood, but how do you then take a mechanism of action approach to uncover deconvolute targets? So a very different approach to upstream target identification to kick off a program. And then from there, we'll move into small molecule hit finding in episode 4. So to everybody who's joined us today, thank you for your engagement. Really appreciate the time, and see you next time. Talk to you soon.